We all know this paradigm that, all of our cells have the same amount of DNA. They possess, therefore, all of the necessary information to make everything that makes us and makes every animal and every plant what they are. And yet, there's a high degree of specialization that takes place that allows cells to really occupy a particular, type or a particular state over others. And the question is, in my mind, is how do you regulate this? Whether you do that for homeostasis, for physiological turnover, or whether you do that for injury repair and regeneration. And so what I would like to do today is to actually give you a sense as to, why is it that we continue to pursue fundamental research in stem cell biology, and particularly on the benefits of trying to really double down on foundational understanding of what is it that regulates potency. And I'm going to start with the following. Because as you all know, we are now bombarded, and in fact, we are adopting and adapting to a phenomenal growth, exponential growth of methodologies and technologies that allow us, biologists and life scientists, to do science that was just not imaginable a decade or even 20 years ago. And there's all talks about, artificial intelligence and the like, but I just want to set the format a little bit so we can really understand where I think we are in this present day. And I'm going to start with natural intelligence. And this is, of course, represented by two folks right here, Charles Darwin and Alfred Wallace Russell, both of whom, by using their brains and collecting large amounts of disparate data that were not related to each other, were able to produce this concept of natural selection, which is fundamental to our understanding of the progression of development, the progression of disease, and of course the progression of species, including our own. This has now, it's not superseded, the attention has now begun to focus a great deal on artificial intelligence. And this is a crazy drawing from DALL-E. I asked pluripotency stem cells, and this is what it drew. Okay, so it doesn't mean anything, but that's what it did, okay? But what I'm saying with this is that you have a new way of parsing data. Like, sort of like, they call it a latent space in artificial intelligence lingo, where you put all this disparate data, then you have an encoder that looks at this data, and you have a decoder that gives you options, gives you hypotheses, gives you answers to questions, right? And so that is what people refer to as artificial selection, artificial intelligence. But what I'm actually trying to suggest is that the information that we're actually feeding into AI is actually artificial data. What do I mean by that? It is primarily work being done in unnatural circumstances, tissue culture particularly. Cells are going to behave in a particular way in tissue culture. And what you're going to learn from that is essentially what do cells do under artificial selection. So I think that the brunt of the work that's being done today in bringing AI to the life sciences is really based on a wealth of information based on artificial selection processes occurring in the laboratory conditions, primarily in tissue culture. And PubMed is a really good example. I think about 70% of the papers there are primarily in vitro or organoids or what have you, and about 20 to 30% is really whole organism data. So that will bias already whatever large language model you're going to apply to analyze this data. Just something to keep in mind, because I don't think it's a panacea that we all wish it were. So what do I mean by this? So if we only look at the organisms, forget about the tissue culture, we just look at the organisms, right? One of the first things that we do when we want to study a system or a model system is that we have to domesticate it. The genetics that we understand is the genetics of domestication. There's not a single organism out there in the wild that's been studied, its genetics been studied in the wild. We bring them into the lab, we cross them, we lower variability, we inbreed them, et cetera, et cetera. So the genetics that we understand today on the 21st century is the genetics of domestication. And it's based essentially on approximately 7 different organisms. And you recognize all of them here, and I'm sure many of us have worked with these organisms at some point or another, or even currently in our present laboratory environments. Now, all of these organisms that I share with you fall into 2 categories. 2 invertebrates and 5 vertebrate animals. So the vertebrate animals shown right here, is essentially 1 phyla. This is an old graph. There should be 3 more phyla added to this. New phyla are being added on a regular basis. That's how little we understand these relationships. And then, this has been the brunt of our understanding of our own biology and our own processes, signaling pathways, cell-cell communication. A lot of the understanding that we have today comes from the study of these systems. Now, the workhorses of invertebrate biology initially selected not because they were developmentally or evolutionarily really important, but because they were really easy to rear, and they had exaggerated some particular attributes. In the case of C. elegans, transparency. In the case of Drosophila, that they were prodigious progeny generators, resulted after phylogenetic analysis to be actually more closely related than people had anticipated. They actually belong to a single group of clade of animals known as the Ecdysozoa, and this is again, a very small sliver of what's out there. Now, all of these systems have garnered a great deal of attention, multiple Nobel Prizes, so it's not like this work is not useful. It is very useful, but it's not all the work that we need to do to truly understand how our own biological processes may have actually arisen, how plasticity is defining those processes, etc. So there are 33 different phyla. As I said earlier, many are being added on a regular basis, and that represents about 9 million organisms that we know. And so if you just do the math, these 7 organisms that we've been studying for the past, almost over 100 years, represents 9 times 10 to the minus 5% of all of the known representation out there. So I don't think we even know what is possible in biology. We know what is possible in these systems, but we really don't know what is possible in the wealth of biological information that's out there. Now, I understand the practicalities. You cannot really work on every organism, but I think we're getting to a point where this actually becomes less and less difficult. Now, I'm going to give you some examples in the next few slides. So why am I telling you all this? Because I think, every so often it's important for us to actually check our premises and ask, are we on the right path to really understand the questions that we want to understand? Or do we need to change, right? And so one of the things that we all know is that the life sciences really reward, beautiful, very simple explanations of very complex systems, right? So when we can distill complexity into a single or a simple principle, that is really a quantum leap in our understanding. It is actually the way it's been since the life sciences have been receiving Nobel Prizes in the late 19th century, beginning of the 20th century, for example. But we also know that a lot of these simple explanations also lead us astray. Some are right, but they're not always right. And so this is a problem that we sort of like brush under the carpet. And I think the reason for that is because evolution does not really follow this kind of simplicity. And so we are the result of evolution. And if you look at it, and if you talk to any evolutionary biologist or any population geneticist, they will tell you that evolution does follow a very different rhythm. It really goes to the beating of its own drum. And it's composed of complex and contingent histories, some of which we know something, some of which we know nothing about. And it's all shaped by the vagaries of time, place, and environment. And there's been no full integration of these processes in our interpretation of our data in the lab. I always wonder, how much natural history people working on Drosophila know about how Drosophila actually live in the wild, or C. elegans, or xenopus, or zebrafish, or even mice. What they know and what we know is how they live in the lab. That's not how they evolved. Their genomes were chiseled by all of these processes that we're not measuring. So it's a great deal of stuff we're not measuring. And therefore, I think it's impoverishing our ability to really make substantial progress in understanding those processes. So I think that simple laws with predictable outcomes cannot really fully describe what I refer to as the procession and fortuitous nature of life. It is a really complex process. And we have reduced it to such simplicity that we're really ignoring many of the facets that will help us eventually understand what we're seeking to understand. So I'm going to give you a couple of examples by what I mean by this. I'm going to start with the simplest eukaryote. These are protozoans, just to give you a sense of what biological space we're not measuring, okay? So everybody knows Tetrahymena. It's a microscopic organism. It's received several Nobel Prizes. The first epigenetic markers were discovered in this organism by David Alice, and the ribozymes were discovered in this organism. So there's a lot of information that is applicable to us that came from a simple protozoan. Now, if you ask AI how big are protozoans, it'll tell you that answer, 40 to 48 microns, right? Because if you look at the data, it says that almost all of these organisms are small. But if you really, really look at the literature, what you find out is that there are protozoans that are about 20 centimeters in size. They're not microscopic. They share common ancestry, and this is the genotypic phenotypic space that a unicellular organism can occupy. So this flies in the face of assumptions that we're making that are not really, supported by the evidence in nature. This is a 25,000-fold difference in scale, okay? That's protozoans. Let's bring it to something closer to our heart. Let's look at vertebrates. In fact, let's focus even more and go just to mammals, okay? So mammals, we all know they're monophyletic. They all share common ancestry. There's no argument in the literature about this. We have enough fossil records to know that all mammals came from an ancestral species, okay? Now, mammals alone differ close to 1 million-fold in size. So the smallest mammal that I'm familiar with is this Etruscan shrew from Madagascar, Suncus etruscus. It's about 3.5 centimeters in size. I think that is generous because it's including the length of the tail, so it's probably smaller if you think about it. And then, of course, we have whales, which you actually see from your window here sometimes, like humpback whales, for example. Those things are huge. They're about 20 meters in length, right? So not only do they differ 1 million-fold in size, but they also differ 100-fold in maximum lifespan, okay? And so how can a single origin produce so many different diverse varieties of mammalian representation? So one would say, well, the genomes are different, okay? Well, it turns out that's not true. The size of the genome of the Etruscan shrew and the humpback whale are pretty similar. It's about 2.5 gigabase pairs. So it's not the size of the genome. It's something else. And we're missing what that's something else. We're not measuring that. And that's just for us. That's just us mammals. So when you actually ignore all this information, when you take away all of this diversity, you're very likely going to compromise innovation. Because it is in those differences that we really make that quantum leap in understanding, what is it that makes life tick. And so, so anything that breaks the rules should really be studied in great detail. So what I'm trying to do in my lab in very small scale is that I want to take the tools that we have today, including artificial intelligence, and try to generate the necessary latent space and data set that will allow this powerful tool not to study artificial selection, but to study natural selection. And I think that if we can do this at some level of success, and I'm using AI for this, I think Charles Darwin and Russell Wallace will be pretty happy. I've never seen Darwin and Wallace smile, so I was really taken aback when I ran this animation because they always look so stern and serious. But that's the kind of stuff you can do with AI. It's fun. It's fun. I mean, I'm really not saying that we shouldn't use it. I mean, it's just a potentially great tool. So how do we do this, okay? So I'm just one lab, okay? But I think the prowess of our technologies today really allow us to do things that we just could not do 10, 15 years ago. So I'm only going to talk about 2 of these 4 animals that I have on this slide. I'm going to talk about the snails on top and planarians in the middle. But we're also working with organisms like Tychodera flava, which is a hemichordate that lives in the Pacific. We've not domesticated this animal. And then we're also working on killifish, which is a short-lived fish and reaches adulthood very quickly. And I'm interested in adult regeneration, so it is the vertebrate system of choice in my lab. So what about these snails? So these snails are originally endemic to Brazil. They invaded Europe and Asia through the importation of tropical plants. The eggs are actually very hardy, so they travel in the soil. And then when they landed in Italy, they just began to eat all of the Italian lettuce. So the European Union wants to get rid of these snails. But what these snails do, which is really remarkable, and we knew this about snails since Charles Bonnet did experiments in the late 1700s, is that they have remarkable regenerative capacities. And so if you can remove an eye out of this snail, essentially in approximately 20 days or so, the eye regenerates completely. And I'll tell you why this is impressive in just a second. We have planarians, which as Evan mentioned to you, these animals can be sliced and diced in a variety of ways, and each fragment is capable of regenerating a complete animal. So this is a remarkably powerful system to study regeneration. And then again, it's a very short process. It takes about a week for an animal that's been decapitated to regenerate a complete head that is functionally integrated to the pre-existing tissue, another fantastic feat of nature. And then we have hemichordates, which are closely related to us, and they were not believed to regenerate, but this particular species, when decapitated, will actually regenerate a head as an adult. So this might be a really, really good opportunity for us to understand how a deuterostome, central nervous system, can be regenerated in an adult. We can't do that, but these animals, part of our ancestry, can actually do it. And in fact, the genes that are being deployed to form the heads in these animals are indistinguishable from the gene regulatory networks that produce the anterior-posterior axis of us during embryonic development. And then finally, the killifish, you know, this is indistinguishable in many ways to zebrafish. They regenerate their tails, their hearts, spinal cord, etc. But we're studying the regeneration of the tail for a number of reasons that I won't have time to share with you today. So let's talk about this snail for a second, because there was no tools for this system when we first ran into it. I want to use this as an example of how relatively straightforward it is to bring something that we know very little about and bring it to the 21st century in a very, very short period of time. It's not going to take a lifetime or a career to actually be able to do this, and it's getting simpler with every passing day. So this is Pomacia canaliculata. As I told you, they have these eyes, and they regenerate well. What is really remarkable about this is that these eyes are camera-type eyes. They are our eyes. They have a cornea, they have a lens, they have a retina, they have an optic nerve, and all of this stuff regenerates in the adult from scratch. Now you have other systems like axolotls and salamanders that may regenerate the lens or the retina or zebrafish or xenopus, but they don't regenerate the complete eye. And so here's an opportunity for us to try to understand how a complex sensory organ can actually be built from scratch in an adult environment in the absence of an otic placoid and all the developmental processes that were required for the formation of this eye. And this, by the way, was the work of Alicia Corsi when she was a postdoc in the lab. She now has and is running her own lab at UC Davis, where she's going to continue developing the system for us to understand regeneration in this system. So here's the types of eyes that exist in nature. And one of the most profound findings in Evo-Devo was the fact that the eyes that had been studied, they all share common regulatory pathways, particularly a gene known as PAX6. So what we have is a situation where so we have simple eyes like the ones we see in planarians. And then we also have a situation, let's see, yeah, in planarians right here. And then we have the compound eye of Drosophila. And then, of course, we have these camera-type eyes, which, snails share with those mammals, particularly humans. And we know that at least for the human eyes, the planarian eyes, and the Drosophila eyes, PAX6 is a fundamental transcription factor responsible for the formation of these eyes, even though their architecture is different. Now, we do not know if that was the case for a snail, so we wanted to test that. So we needed to develop tools for these things. I'm going to give you one quick example of how we do this. We have no markers. We have no transgenesis. We have no antibodies. We have no markers. But we have things that label DNA because these things have DNA. And so to sort out and understand the cell types that are present in these animals, we were able to leverage AI to help us classify cells on a number of attributes that we could easily measure through flow cytometry. So what we would do is that we take these animals' cells, make a single cell suspension, pass it through a flow cytometer. Each cell that passes through the flow cytometer is photographed. We collect that data, the DNA is labeled with a DNA dye, and then we generate a large collection of images, and then we can process these to ascribe to these individual cells the specific characteristics. We can use principal component analysis to separate cells based on site scatter and the like, and then as we do this, we increase the granularity of the details within each of these clusters, and so we can look at the bright field image, the site scatter, the DNA labeling, and then merging all that, and that's cluster 1, for example, and then all of these clusters that were turning out to be different receive a very specific set of attributes. We can take all of these attributes and then visualize them in the clusters by their cell size, their cell circularity, granularity, and stuff. This is the data that you need to generate in order to train a large language model. And so we generated all this data, feed it into a language model, and then allow it to tell us, whether or not it can actually recognize cells without even knowing what those cells are. And when we do that, essentially what we produce now is a trained a neural network that when it sees the data, it can actually sort the cells without even knowing what the cells are, just based on the attributes that they have. So we're able to take all of the cells of the hematopoietic compartment of these animals and separate them without having to develop a bunch of antibodies to separate the subclasses in there. And that's what you can do today. And so you don't really need to have all this infrastructure that was required for these other things. You can actually just use these kinds of measurements to allow you to produce very, very specific. We can sort these cells now, subject them to single-cell sequencing, and further begin to decorate them with molecular markers, et cetera, and begin to dissect them, right? So this is something that can be done with any system that has absolutely no, no tools available for them, okay? So the other thing we needed to do is, can we genetically modify this system? Because at the end of the day, we need to introduce functional analysis into these guys. So Alice was able to get these cells to undergo in vitro fertilization, which you can see right there. And then we were able to inject RNA into these developing embryos, and saw that actually the mRNA is translated into a protein by measuring GFP presses, right? So it is possible to manipulate this in the ways that we normally manipulate other embryos in the system. So what we thought we would do is that we would try to use these methodologies to introduce CRISPR-Cas9 into this system, and then begin to do crosses to see if we can actually do Mendelian genetics, and then see whether or not we can actually detect functions for these genes. So we injected animals with CRISPR-Cas9 guides against PAX6, generated heterozygous, as the cross is a very simple cross, and then we looked at the mutants. And what turned out to be the case is that PAX6 is absolutely required for the formation of the eye in this system, okay? So that's proof of principle. I will tell you, it took us longer to get the permits to bring the snails into the US than to develop all this system, okay? That just shows you, what's going on, right? And so I'm very excited that this is a tremendous opportunity for us to really go and explore new biology. Biology that is, we always assume is out of our reach because the methods are not there, et cetera, et cetera. I think that gap is diminishing at a very, very fast rate. And it will open the opportunity to, generating tremendous amounts of new information. So what we want to do, we just recently introduced a fifth system, this jellyfish right here, that undergo this this reversal of lifespan. So they go actually from adults to polyps if you injure them. So they revert developmentally. And so again, we want to study all of these organisms using all of these methodologies with the idea of being able to generate all of the data that might be required for us to drive a conceptual advance in understanding how adult tissues are being regulated in response to injury, in response to regeneration, and how that plasticity may or may not be driven. So that's the goal. I'm 61, I'm giving myself 9 years to try to do this, because by 70, I'm not sure I'm gonna be able to keep up with all this stuff, okay? So, but I think it's a very exciting time, even though, the prevailing winds would argue differently, but I think that it's actually really exciting times for us to really start thinking about what might be possible in biology today. So I want to tell you, with the remainder of my time, what we've been doing with Planarians, which is the most mature system we have, to help us understand stem cells, the agents of regeneration, as well as the somatic cells that support those regeneration agents in Planarians. So Planarians essentially can be split into like two general cell types. There's a population of postmitotic senescing somatic cells, neurons, muscle, gastric cells, epidermal cells, etc. And then there's a large collection of negligibly senescent, self-renewing pluripotent stem cells, okay? And so when the interaction between these two different types of cells is what actually produces the Planarian and what actually results in all the biological properties that are displayed by this organism. So a few years ago, Blair Benham-Pyle, when she was a postdoc in the lab, decided to try to understand the cellular composition and the dynamics and transcriptional dynamics of the smallest possible fragment of a Planarian that could actually regenerate a complete animal. That turned out to be a biopsy of about 0.75 millimeters in diameter, consisting of about 8,000 to 10,000 cells. We would take that biopsy, we would let it regenerate, and as you can see here, in 14 days, from this little chunk of tissue, you end up producing a complete animal that is moving around, is eating, and the like. So what we did essentially was to take these developmental stages, these regeneration stages, dissociated them into single cells, subjected them to single cell sequencing, and generated a map of about an atlas of about 300,000 different cells with the corresponding transcriptional profiles. This was an extremely illuminating collection of data that we're still mining and Blair is still mining. Again, it encompasses 300,000 cells, and then it identified 90 different transcriptional states from 8 tissue classes. So there's a lot of transcriptional states, even in the 8 different cell classes that actually give rise to the complete animal. What we learned thus far from studying the processes of regeneration and studying the stem cells of these animals is encapsulated in the following points. So we learned that the most undifferentiated cells in planarians express large amounts of Pee-wee transcript and large amounts of Pee-wee protein. We thought that by purifying those cells, we would really purify the stem cell, but this is what 8,000 cells of high Pee-wee content look like. You can actually deconvolve them into at least 12 different groups. And then we found that there's one particular group, the NB2 category right here, that is something that we have not found before. We think this is actually the stem cell in these animals. So we know that also the somatic cells appear to display transient and functional augmentation of their activities during regeneration. They're not static bystanders. They'll actually change their expression profiles, and I'll show you the evidence in just a second. So knowing that the stem cells themselves can occupy multiple cell states in both homeostasis and regeneration, and also knowing that the somatic cells, which are supposed to be terminally differentiated, can actually also change their expression profiles, posited a really interesting paradigm for us. So what we found, for example, in the somatic cells is that within the muscle cell class, for example, we find a very small subset of cells that will actually change their expression profile to turn on a number of genes like collagen, NODUM, and a few others. We select those genes for RNAi, for silencing, and when we do that, what we see is that these deletions of function have an effect on the polarity of the tissue that's being regenerated. That's for mesodermally derived tissue. We look at ectodermally derived tissue. We also found a subset of cells in the ectoderm that is activating a number of genes, a relatively small number of genes. We targeted all those genes with RNAi, and what we find is that for regeneration, it only affected stem cell proliferation and nothing else. And again, when we look at the endoderm, we see that there's also a collection of genes that reaches an apex or a zenith, so to speak, somewhere in the middle of regeneration, and then their expression decays until it's no longer detectable after regeneration. And if you interfere with the function of these genes, this actually affects stem cell maintenance and the remodeling of the tissue that's required for the animal to establish de novo their AP, DV, and mediolateral axis, okay? So the fact that there is a small subset of cells that can actually change their expression profile, and that minute change has this profound effect on regeneration, tells us something really, really important about why is it that these animals may be so good at regeneration. We refer to these cells as tracks for transient regeneration activated cell states, and that we now also have evidence and published it, but I don't have time, to show that they actually also exist in vertebrates. We found them in the regenerating tail of the killifish. And that paper has been published, and if you want to look at it, you can see what I mean by that. Okay, so how then do the stem cell and somatic cell compartments interact in order to process all this information to produce form and function? Especially when you're just removing a tiny bit of flesh, and from that little flesh that's randomly selected from the animal, you get to reset all of the polarity as such. So we generated a spatial atlas of Planarian regeneration using SLIDE-seq v2. This was our pandemic project. It's about to come out, it's no longer under review. It's in press, and it's about to come out. But I'll give you the summary of the work. So we essentially took an animal, removed the tail, allowed the tail to regenerate for 6 hours and 48 hours, embed these in OCT, section them. It looks almost like a little collection of flower with petals, and then use one of the sections for H&E that allows us to see whether or not we collected the tissues we're interested in. DAPI allows us to count all of the cells that are in all these sections, and then the SLIDE-seq v2 will give us a transcriptional representation of the cells that landed on the barcoded beads that are present on the puck, right? So we apply the same kinds of methodologies that we use for single-cell sequencing, except that this is just for beads. And you can see here that all of the major cell classes that are found in Planarians, all 8, were represented in all these sections. We can take now all of these beads, generate UMaps, and now look for clusters of beads, okay? And so we're going to focus on clusters 26 and 17, which are present at the bottom of this UMAP right there, because these beads had something really peculiar to us. We're trying to find out what surrounds the stem cells, okay? The beads are larger than the stem cells, and they're larger than most cells. So we're looking for beads that had a mix of stem cell-specific expression profiles and other cell type expression profiles. And so when we do that, we find that there's a large collection of beads that really gave us neoblasts surrounded by other cells, and we want to know what those cells were. And so we found a new class of cell that had not been described in planarians before, possibly because they are so big that they were not sorting through. They were not going through the filter. And Biff Mann, who is the postdoc who did this work, refers to these cells as Hecatonoblast because they have a lot of tentacles. And they are illustrated here by purple, and they are marked by a matrix metalloprotease 1. And what you're going to see here is the purple is the Hecatonoblast, the green is the stem cell. And you can see that the difference between 6 hours and 48 hours post-amputation is that there appear to be more PW positive cells, the stem cells, in close association with the purple cells, which are the Hecatonoblast. White were the nuclei labeled by DAPI. So we thought that maybe, just maybe, this might be the niche. This might be the cell that helps support the function of the neoblast. So we went and purified the cells, found a transcription factor that is required for the formation of these cells, delete the function of this transcription factor, and it had no effects on the stem cell regulation. So it was really a false positive. And so then we decided to look at the actual environment in which these cells reside. And so we did a series of serial electron microscopy analysis to identify what cells were in direct opposition or in direct contact with the stem cells. So here's two stem cells from this quadrant X right here in close opposition to each other. But if you notice, there's no cell-cell contact between these two cells. We looked at Y. This is a Hecatonoblast right next to a neoblast. And you can see also there's space in between the two cells. There's no physical contact. And then we look at this other one, a larger Hecatonoblast and another stem cell. And again, there's distance. There's no direct contact between these two cells. I found this to be extremely puzzling. So what we did then was to take all of these sections and reconstructed them to be able to see whether or not this was a common event or it was just that we just selected the wrong cells for analysis just by randomness. So here are all the serial sections, and what we're going to do now is that we're going to take all of the serial sections, reconstruct them, and then again using AI, we're going to look for all cell-cell interactions that occur in this particular cohort of cells. And so here's what the movie looks like. In the center is the neoblast. This is the clear area, and all of these things that are coming out are the different cells that are right next to the neoblast. So we took about 200 of these and generated the following maps. So the lines that you see here indicate a clear physical contact, either a desmosome or some junction that is present between these cells. N is the stem cell, the neoblast. You can see that in this particular cluster, there's no context surrounding this neoblast. And so after measuring 200 such neoblasts and their environments, and then measuring all the cell contacts that were present in this situation, what we find essentially is that all neoblasts examined display an absence of detectable cell-cell junctions with their neighbors. Again, this is very bizarre. This is usually not how you find stem cells interacting with their environment. But again, you know, these are also incredible exaggerators of potency. So there may be something really important that can emerge from this. So if they're not talking to each other by direct cell junctions, where do the extrinsic signals come from? So we know they come from a distance, and we know they come from a distance based on the work that both Blair and graduate student in my lab, Aubrey Kent, were able to do, which is that right upon amputation, what we find is that there's two tissues that really respond to amputation very, very strongly. The stem cells don't appear to respond immediately to amputation, but the epidermis and the intestine do. So we really understand the epidermis the best in Planarian, so we thought we're just going to focus on the epidermis, okay? And then see how this tissue is really responding to injury and how it may actually be affecting the stem cells. Now this brings me to the following observation, which was made by Brett Pearson when he was a postdoc and we were in Utah, which is that we found that the really peculiar finding that P53 in planarians does not need to receive a genotoxic stress to be activated. It is on all the time. and is only expressed in about 22-23% of all of the neoblasts, and in about 100% of the early progeny, post-mitotic early progeny of these neoblasts. And at the time, what we thought was happening here is that p53, we thought, was regulating potency by essentially promoting the differentiation of one of the daughter cells from the stem cells. Because when we deleted the function of p53, we never got any progeny. We only got symmetric cell divisions, and only got more of these neoblasts, and eventually they become exhausted, and the animals would eventually perish. So we concluded at the time, this was in 2010, that p53 was controlling proliferation by promoting differentiation. But now there's a lot of data to suggest that p53 may actually be regulating, at least we think that's what's happening in planarians, oncogene-induced senescence in post-mitotic cells, which apparently is actually a mechanism that is being used for regulating aging in some of the differentiated cells of organisms that are constantly renewing their tissues. Okay, so that suggests to me that there is something really interesting about the way in which stem cells and their progeny are being regulated. So this progeny, this early progeny and late progeny of the stem cells, we have studied in great detail. So we have specific markers for each of these developmental stages, from the neoblast all the way to the differentiated cell. Genes like PROG1, AGAT1, CPUF, transcription factor, and rootletin. And it is probably the cells that have the fastest rate of cellular turnover in these animals. Okay? So we're going to focus on these Agat positive cells because Aubrey obtained a really interesting phenotype. And when we targeted these genes by RNAi and we measure what would happen to the neoblast, to the stem cells, in the absence of this function, what we found is that one of the Agats was able to actually change the mitotic profile, or the mitotic numbers, of a neoblast when Agat1 was RNAi treated, and that was Agat2. The number of mitosis dropped significantly when Agat2 was targeted. Okay? When we looked at cell death, we also found that the elimination of Agat2 increased apoptosis. So we have a situation where a gene that is expressed far away from the neoblast in a very specific cell type is actually having a significant effect on the neoblast themselves. So there is communication at a distance. We just don't know how that is actually mechanistically deployed. When we purified the Agat1 cells and subjected them to even more sequencing analysis, what we find is that these cells have both a metabolic and transport signature profile, expression profile, which is very interesting because these are supposed to be transitional cells that are moving from one developmental stage to a terminally differentiated state, which is the epidermis. So why would a cell that is on its way to becoming epidermis would have such well-defined transcriptional profile? So here's Agat positive cells. They are making a lot of metabolites, and they're also expressing a large number of transport and trafficking molecules in them. So where are these cells? So you can see that they're essentially dispersed all throughout the edge of the animal. This is a cross-section of the animal. The brain is here, the pharynx is here, and you can see that all of these cells, expressing all of these Agats, are on the periphery of the animal. The neoblasts are somewhere here in the middle of the animal. Okay? So somehow, at a distance, these cells can actually perturb quite effectively the activities of these neoblasts. So these Agat-positive cells are supposed to produce creatine. And now we know it's happening because it supports regeneration, because Agat is involved in the generation of things like homocysteine and creatine. Okay? So what Aubrey did was to target this gene by RNAi, and then here are the phenotypes. You can see that the animals are incapable of fully differentiated their heads, and eventually they lyse. So here, they fully differentiate their heads, they lyse. And the intact animals eventually just lyse and die. If we now supplement this RNAi background with creatine, what she was able to find was rescue. So in fact, these cells are making creatine, and the creatine is required for the neoblast to do what they need to do to launch a regenerative response. Okay? And so this is actually a very peculiar finding in my mind. Because again, these are late progenitors. There was no function ascribed to them. They're supposed to go on and differentiate into something else. And so what we are suggesting now is that we need to change the way we look at these cells. So the previous view for us was that this was a transitional state into epidermis. And Aubrey's findings suggest that these cells are involved in metabolite production. They affect regeneration. They affect stem cell maintenance. And they also regulate tissue turnover. So what I'm thinking is that this particular collection of cells may actually represent a metastable organ, some sort of endocrine organ that is represented by these cells, and it allows for communication and integration of function into these animals. And these animals being acoelomates and having lost a lot of organs apparently, or never having them, suggests that maybe there may be cells out there that really don't congregate into a specific organ, but they're actually playing an endocrine function. Okay, so how is this happening? So this takes me to the last part of what I want to share with you. This is also a work in progress. Some of it is under review, but I'm going to share with you. And it's the work of one of our postdocs in the lab, Anand Vidyanand. And so Anand is very interested in understanding cell-cell communication, and his work led him eventually to look at exosomes in planarians. And this is from a review by Randy showing what these exosomes look like. And I think most of you know what extracellular vesicles are, so I'm not going to belabor the point here. So we look for these exosomes in planarians and these extracellular vesicles. We find evidence by electron microscopy they're there. And we also find that they're being secreted by the animals, and they're positive for CD9 and CD63, which are specific markers for these EVs, okay? We purify them using methodologies that have been accepted by the community at large, and we found that in fact we can separate large EVs and small EVs based on their protein composition. So we did proteomics on these purified EVs, and you can see that on the left are the small EVs, on the right are the small EVs, and on the right are the large EVs. And we're going to focus on the small EVs. And so the reason for that is as follows. So we have an animal we have not fed. We give them a beef liver paste. This is the 150 that we spend on food for these animals. And then we're also going to give them food that is laced with double-stranded RNA. And we find something really peculiar. And that is that if you only feed them liver, you get a single peak of production of EVs. This is 10 to the 6 particles per milliliter. And that's in gray. It goes up and it goes down and it stays down. But if we provide the liver with RNAi or double-stranded RNA, you can see that there's one peak that coincides with just food, and then a second peak of EV production that occurs later on days 3 or 4. This is actually very interesting because it suggests that double-stranded RNA is doing something to generate this EV production. We can label the double-stranded RNA with the juxtagenin or some other fluorescent marker, and we can see how it appears to be encapsulated in some sort of a granule inside of the cells, inside of the animal, sorry. So what we did then was to repeat the experiment, wait for 2 days after feeding, amputate the animal, and then wait 2 days after amputation, and then isolate the EVs, treat the EVs with RNA, and then subject the EVs for imaging and for a small RNA sequencing, okay? And so what we find essentially is that the RNA, that the EVs have RNAs in them, and they're not on the surface. They appear to be inside. They appear to be cargo in these EVs. But the more important thing is that we actually found the siRNAs of the gene that we're targeting by double-stranded RNA encapsulated inside of these EVs. And that's what these reads are. So this is our control, ONC22, and this is a gene that is expressed in planarians, OVO, which allows for eye formation. And you can see the millions of counts right here, suggesting that all of the siRNA that is being made from double-stranded RNA somehow appears to be encapsulated in these small EVs. So if that is the case, we may have figured out how is it that RNAi is so efficient in planarians. It's probably because of these EVs. So we did the next obvious experiment. We'll repeat the experiment, we will purify the EVs, and now what we'll do is that we'll inject these EVs into a naive organism and see if we can actually trigger the expected phenotype that siRNA by feeding would be produced. And so we did that for OVO, and as you can see here, when you inject EVs that contain OVO siRNAs, you can see that the eyes don't regenerate. And then when you inject EVs that have processed beta-catenin double-stranded RNA, after amputation posteriorly, the posterior end begins to regenerate what appears to be a head, suggesting that these EVs are really integral to the transfer of siRNA inside of these animals. So I'm going to conclude with what I want to share with you today. So we think that planarian stem cells do not appear to have cell-cell junctions with their neighboring somatic cells. That there is long-distance regulation of stem cell dynamics and is a predominant form of extrinsic regulation in these animals. We think that these transitional epidermal lineage cells may be key regulators of stem cell function and may represent a novel endocrine organ in planarians. It's a different way of thinking about what an organ may actually be. And that extracellular vesicles appear to be involved in affecting RNAi and are likely common vehicles for the transport of other biomolecules in planarians, for that matter. So the last thing I'll leave you with is a perspective, okay? And which is like, why is it that planarians are so long-lived? I mean, I've had these animals in the lab since 1998. It's a clonal line that was produced from a single animal. We have millions of these animals, and we see no evidence of senescence. The line continues to grow exponentially, okay? And so the way we think about aging in mammals is that things become really, really bad with disease incidence at or about 60. I'm 61, so I'm getting ready to experience all this. I'm not looking forward to this, but that's what the data says. And what people are arguing is that our bodies, or evolution, has actually done the following, which the natural selection has pushed senescence all the way to the end by preventing, senescence in the earlier years to allow for, sexual maturity and reproductive fitness. This is the work of James D. Gregory in Colorado and a few others that ascribe to this particular hypothesis. So this is what we see. So the lifespan for us is about 80 years if we're lucky. The going hypothesis is that natural selection is essentially repressing senescence, and then that repression sort of like gets loosey-goosey around 60, and then everything just goes to pot. And here we have planarians, which have an indefinite lifespan. I mean, they just live for a very, very long time. And what I'm beginning to think is that it's just the opposite. So I think what's happening in planarians is that instead of slowing down senescence, they are promoting senescence. And so by inducing really rapid cell turnover, so the cells that become post-mitotic are not going to live for a very long time. They actually die very quickly. And so that provides very strong selective pressure to retain a constantly self-renewing pluripotent stem cell population, which may explain why planarians have these crazy adult pluripotent stem cells in their anatomy. They've been selected for this. And so the perspective as to why they are so long-lived, so we think that this rapid cellular aging, which is represented by p53, for example, it just induces senescence quite quickly. And this turnover actually does place this strong selective pressure on organisms to preserve these adaptable stem cell reserves. And so rather than prolonging somatic self-lifespan like we do, and the same thing with flies and nematodes, evolution in planarians has favored faster senescence and is heightened the dependence on stem cell-driven renewal. So this is a massive exaggeration of this process, which suggests to me that we should be able to reveal a great deal of what is it that drives senescence in other systems by looking at planarians in general. I think that if that is the case, in order to sustain this, the niche has to be different. So having a niche where it's really dynamic, that is not buttressed by cell junctions, that is working at a distance, that is very plastic, may actually allow for the cultivation of these pluripotent stem cells. Of course, these are all conjectures and hypotheses that need to be tested. But in sum, I just want to thank all the people that make the work possible. I mentioned their names along the way. This was the lab in 2023, and this is the lab in 2024. And then, of course, all of the support that we get from our institute to make all these experiments fun and exciting. Thanks very much for your attention.
Thanks. Beautiful talk. I'm wondering about the P53. And probably you get this question a lot because those of us that are like working in mammals think a lot about P53 as mostly expressed in cells where there's genotoxic stress.
Yes.
Do you think that its ancestral role is to promote differentiation, and then later it picked up a role in the presence of stress?
Yeah, no, that's a really, really good question. I think I favor the hypothesis that the ancestral function of P53 is really to promote differentiation. And the reason for that is based on what we know evolutionarily about P53. So choanoflagellates, which are single-celled organisms, okay, they have P53. But not only do they have P53, they already have a copy of P53, which is either P63, P73. It's difficult to distinguish the two, but there's already two copies of this gene in a unicellular organism. If they are involved in genotoxic stress or in preventing, tumors or being involved in tumor suppression, I don't know what a tumor looks like in a single cell organism. So I don't think that holds water, right? It didn't have to work out that way, but that's the way it worked out. And if you look at other single cell organisms, you find that a lot of these molecules that we normally associate with either oncogenic activity or tumor suppression activity, they're already there. And in many cases, they're duplicated. So that appears to suggest that these genes have actually a propensity to evolve novel functions. And so, so I think that p53's function in ancestral systems were essentially to promote differentiation and in turn promote senescence. The experiment that one would have to do is to essentially try to, if we really understand this, we should be able to allow p53 to turn on in mammalian cells without triggering apoptosis, for example, without triggering a response. And the last thing that I'll say about that is that p53 being like the guardian of the genome or genome integrity and so forth, that may also be part of it because we think also that the cells that are being selected for differentiation out of the stem cell pool are very likely accumulating mutations, and those are the ones that go into terminal differentiation, and they put them to work instead of subjecting them to immediate apoptosis. So I think the story is going to get richer the more we look at other systems and how these particular functions are executed in different evolutionary contexts. But yeah, that's what I favor right now.
Thank you so much for a very interesting talk. So I have two questions. One is, so you mentioned that not only the stem cells, but also the somatic cells will undergo some kind of reprogramming for the regeneration. And I'm very curious about what triggers that, like how the somatic cells will turn to that state to support the niche for the stem cell regeneration. And also, my second question is, so you mentioned that you guys are interested in how the natural selection works for the planarians in the nature. So I'm wondering, I think the planarians undergoing the regeneration is very vulnerable to things like predation or something. So I'm wondering if they have any kind of mechanism to protect themselves while regeneration.
Yeah, so those are both great questions. So I'll start with the second one because, we've done a lot of natural history work on these animals to see what happens. So when they undergo, there's two things going for them. They reproduce by essentially cutting a little piece of their tail out, relatively small. It's about 0.5 millimeters in length. And that little fragment essentially becomes immobile. It's completely paralyzed, and they're dark and they're very difficult to see. Okay, so I think a regular fish or a predator will probably not find it. Okay, that's one explanation. We don't see that being the case. The second thing also is that they are prodigious producers of mucus. Okay, and the mucus is very, very rancid and very, very sticky. And I've actually seen animals try to eat a planarian and spit it out. Okay, that's how bad it is. So I think there is the possibility that they're not really tasty, and that and they're very small. And so the predators that exist most frequently in a planarian population are actually protozoans. So protozoans will actually be able to penetrate the animal if the wound after healing, which is actually very fast, about 30 minutes before it's completely sealed, they can actually get in, populate inside of the animal, they eat the animal inside out. And that's usually when we see these populations disappear. And so, that's the situation that we know for normal predation in the wild. Now, for the first question, yes, we would like to know how is it that you can actually modify very specifically a subset of genes to express or change the transcriptional profile of that cell so it can acquire new function. Okay, so I'm sure that the epigenetic landscape is going to matter a great deal here. We haven't really found a way to do it with sufficient granularity to really look at the cells that actually do this. It's a very small percentage of the cells. My suspicion is that with new the advent of new attack-seek methodologies and the like, and as well as AI, we're going to be able to maybe purify them out of the whole population of things. So that we're trying to do that. But that's our first possibility, is that there must be, and then we can do an RNAi screen, there must be some chromatin modifiers that are being specifically activated in a very small subset of cells to drive the expression of genes that were not previously expressed in those differentiated cells. It's very, very surprising. I'll give you a really beautiful example. So DPP is expressed along the midline of the animal, okay? And it's really just a row of individual cells. So if you take a flank of the animal that doesn't express DPP, and you now watch it, because it needs to regenerate the midline, the first thing that you see is a salt and pepper expression of DPP in differentiated cells. It even happens when there are no neoblasts in that tissue. It's not all the cells, but it's just a subset of those cells express DPP. And then eventually, they realign to the edge of what's going to be the future midline, and then a blastema rises on that size, and then the animal begins to regenerate. So there's an exquisite control on the number of cells and possibly the type of cells that are being subjected to this change in expression profile. We just don't know what the mechanism is, but we can definitely measure it. We just know exactly what molecules are affecting this. And Beth Duncan, when she was in the lab, that's what she was trying to do. She essentially cloned all of the histone modifiers, DNA and RNA modifiers, and ran a screen to see if you could identify what molecules may be most likely involved in this. And MLL1 and SET1 are the prime candidates, and that's what she's working on right now.
Great. I think most of the questions that came in online have been covered by the answers here. So I think we'll just thank Alejandro for just a really stimulating talk and wish him well.
Okay, thanks very much, and thank you very much for being here.
And see you all next month.